Today we're going to talk about chromosome variation, but before we can do that, we have to remind ourselves all about chromosomes. So if we pan down, we're going to look at a chromosome. Now, as we can see, this is a condensed chromosome, but this is a chromosome that has not yet replicated. This is evidenced by the fact that it is singular. Okay, there are three things that all eukaryotic chromosomes have to have. Okay, all eukaryotic chromosomes have to have a centromere, and so that is represented by this pinched in region here. Then remember, the centromere can exist anywhere along the length of the chromosome. All eukaryotic chromosomes have telomeres, and I've represented those here by this lovely orange. Pardon the coloring. And then finally, all eukaryotic chromosomes have to have an origin of replication. And remember that most eukaryotic chromosomes don't have one origin of replication, but they have many origins of replication. And so I'm representing these here by this. Now, you usually are never going to see a condensed singular chromosome. It is going to be a blob of uncondensed DNA complexed with the proteins. And so it exists most of the time as chromatin in the cell. Okay, here we have two pairs of homologous chromosomes. This is one pair of homologous chromosomes. This is another pair of homologous chromosomes. This is known as homologous pair. Oops, and this is a homologous pair. We want to keep them nice and neat. Okay, so notice how the homologous pairs have a couple of things in common. They're both the same size. These are the same size, these are the same size. The centromeres are found in the same place. If I had stained these, they would have stained in exactly the same way. Okay? Now, this is the sister chromatid for this. This is the sister chromatid for this. These are sister chromatids, and these are sister chromatids. This is a non-sister chromatid for this. This is the non-sister chromatid for this. So if crossing over were to occur, this is where crossing over would occur. And if crossing over were to occur here, it would occur there. Okay? That's where the crossing over would occur. This is a non-homologous chromosome for this. These are non-homologous chromosomes. All right? And so that's a primer on how we recognize chromosomes, chromatids, and homologous and non-homologous chromosomes. The last thing I want to remind you of is that through meiosis and mitosis, once these sister chromatids separate, once you get separation here, this is no longer a chromatid. A separate chromatid is actually called a chromosome now. Okay, eukaryotic chromosomes are actually based upon the position of the centromere. Now, all of these chromosomes have already replicated. As you can see, there are two strands of DNA in each of them. All right. The position of this centromere means that we call this chromosome a metacentric chromosome. The centromere is in the middle. When the centromere has actually moved slightly upward, this is known as a submetacentric chromosome. As the centromere has moved further up the chromosome, this is now known as an acrocentric chromosome. When the centromere is at the end of the chromosome, this is now called a telocentric chromosome. Now, there are a few things that we also need to remind ourselves about this. The first thing is, cytology reports have shown that there is a small amount of the chromosome even at the ends of these telocentric chromosomes. So that's an important thing to remember. There still is a small amount here, so that there are some scientists who question whether or not we should even be using this classification scheme, although it is still used today. That's the first thing. The second thing is this classification scheme has led to how we report where genes are found on a chromosome. So for example, by definition, when we look at these chromosomes here, the shorter arm is known as the P arm, and P is from the French word petite, P for short, petite. And then the other arm is known as Q. Q doesn't stand for any cute little French word, it's just the letter after P in the alphabet. So the P arm and the Q arm. And so when you see, when you are doing a search for a gene, say in GenBank or in the online Mendelian Inheritance in Man sites, they may actually list 15Q and then another number, and that tells you that if this were chromosome, say 15, that the gene is located on the long arm of chromosome number 15 in this particular area. Okay, in front of me I have two homologous chromosomes. As you can see, one is represented by a beige color, the other one is represented by a brown color. And on it we have a couple of different colors, and each one of those is representing an area on the chromosome. These are not necessarily representing genes per se, but just areas that are homologous to one another. We're going to use this set of chromosomes, or others similar to it, depending upon how badly we botch these up as we rearrange them, to represent the 4 types of chromosomal rearrangements that we want you to be responsible for. Those rearrangements are duplications, deletions, inversions, and translocations. Okay, so first chromosomal rearrangement we want to talk about is a very simple one, and that is a duplication. Duplications can often occur, and in this case what we're looking at is a duplication of the area involving the blue and the green areas. So here's our normal chromosome, and this is the chromosome after the blue and the green area have duplicated. And as you can see, this is duplicated in order. When it duplicates in order, this is known as a tandem duplication. When a duplicated chromosome goes to line up with its homologous pair at meiosis, there's a problem. You have additional material on this chromosome that doesn't exist with its homologous pair. So what happens? The duplicated material loops out so that the regular material can still line up properly, as you can see here. One of the interesting things with the duplicated material, I talked about the fact when it duplicates in order like that, it's called a tandem duplication. Say this had been inverted, right? When that happens, that's known as a reverse duplication. In some cases, the duplicated material may not have actually ended up here. In some cases, that duplicated material may have ended up, say, over here. In that case, it would have been called a displaced duplication. All of these are possible with the chromosomal arrangement we call duplication. The next chromosomal rearrangement we want to talk about are deletions. And a deletion, of course, would be when we lose a piece of DNA. As we can see here, this is our normal chromosome again. And up here you can see we've lost the blue and the pink regions. Poor blue is feeling really picked upon right now. You can see missing, normal. Once again, when pairing up occurs during meiosis, we have a problem because one of the homologs contains more information than the other homolog does. In this case, it is the normal homolog that contains the proper amount of information, but the homolog that contains the deletion is actually missing DNA. So the homolog that contains the normal amount of information will actually loop out that additional information. This is known as a deletion loop because the information that is not matching up has nothing to base pair with, and so to match up with, and so it is looping out. As As you may have noticed, our chromosomes have switched from brown to gray, and that's because I got a little testy. But these things happen when you're dealing with bad chromosomes. These are good chromosomes. So we're now going to talk about our third type of chromosome rearrangement, and these are known as inversions. And we're actually going to talk about two types of inversions. The first type of inversion that we are going to talk about is called a paracentric inversion. Para, that is P-A-R-A, and if you look down at the bottom, it's spelled out for you. Paracentric inversions are inversions that do not involve the centromere. So here's an example here. This piece of DNA, this is our normal chromosome here. This piece of DNA has undergone a paracentric inversion, where the piece of DNA has actually been cut out and flipped. Cut out and flipped. So it's very exciting. And so as you can see, when this is pushed up against this one, it doesn't quite line up, does it? And so we're going to have some very exciting manipulations and contortions in order for these two to line up at metaphase. Okay, with a paracentric inversion, when the chromosomes go to align at meiosis, we have a problem because of the way that the alignment must occur. And so I wanted to demonstrate this, so work with me here. What will happen is the chromosomes, these are very, very thin, so work with me here. We're going to try to make sure that they don't fall apart completely. But this is what happens. The chromosomes will have to loop around here, okay, to form to make the alignments that they need in order to match up, okay? So the looping will occur. Trust me, this looks much better in real life than it does with clay. Okay. Now the really interesting thing that happens when we have these inversions is what happens when crossing over occurs, because that's when things start getting very interesting. So let's assume that we get crossing over between non-sister chromatids, okay? So here's crossing over, all right? So I'm splinching these two here, right? And I'm splinching these two here, all right? Everybody sees what I'm doing, all right? So there's my crossing over. And I'm crossing over between the yellow and the green, all right? So now I'm going to separate up my chromatids at the centromeres. I'm separating my chromatids at the centromeres, all right? So let's follow along with me. Here, this one separates pretty easily. And remember, this is the gray ones. The lighter gray ones are the ones that did not have the inversion. So this one separates, and as you can see, no problem. This has all of the DNA intact and without any duplications or deletions. All right? Now, I'm gonna go from the bottom down here as well. All right, this one here, if I take this one out here, I follow along, look here, just following along here, this has all of the DNA as well. And there are no duplications or deletions here as well. I still have my inversion loop, right? I still have the inversion, but there are no duplications or deletions. So as long as this inversion did not occur in the middle of a gene or anything, this DNA might actually be okay. You don't know. All right, let's follow along here. I come here, I get to my crossover, right? I'm gonna keep pulling my DNA and see what happens. Oops, look what happened here. I have two centromeres. What is gonna happen? It's going to be pulling to two different parts of the DNA, and this is exactly what happens. I end up with two pieces of DNA. You have no idea where the DNA is going to break when it goes to which side of the cell. And so it just broke wherever it ended up. And look at this poor piece of DNA. It doesn't have a centromere. It's very sad. Without the centromere, this piece of DNA is going to be lost, because without a centromere, it's not going to segregate to one side or the other, and it is called, this is acentromeric DNA, and that piece is lost. So this is what happens in our gametes. We end up with one gamete that is completely normal, one gamete that has the total complement of DNA, but does have the inversion. And then you have two gametes with a lot of messed up DNA. So here we are with our increasingly sicker looking chromosomes. And now we are looking at the second of the two type of inversions. And this one is called the pericentric inversion. So this is P-E-R-I, centric inversion. So this is the inversion that includes the centromere. So once again, the gray chromosomes represent our normal chromosome, and the brownish ones represent our chromosome where the inversion has occurred. So our normal one would have the yellow, green, blue, and in this one the yellow, green, blue has been flipped in order. And so once again, we are going to show what happens, how we get the proper alignment during meiosis. Okay, so now we have formed our inversion loop, and again we're talking about the pericentric inversions. So I'm going to do a crossover again, and I'm going to do a crossing over here. So once again, here's a crossover. I'm going to do crossing over between our non-sister chromatids. So I'll splinch these two together, and I'll splinch these two together. There we go. And let's see what happens when these centromeres divide and we make our homologous chromosomes. The chromatids separate and we go to different cells, see what happens. So this first one, we start peeling away, the centromere divides, and here we have a normal chromosome, right? All in order, no duplications, no deletions, looks like a happy camper, okay? What happens, I'm gonna go from the bottom, this one here, and if we peel away here, assuming I don't break anything, we get a centromere. That's always a good sign. Peel away here, and once again we have no duplications, no deletions, and here we have the chromosome that has the inversion, but again, no duplications, no deletions, just the inversions, and that's, relatively speaking, not so bad. Okay, following from one end here, I go along here, keep following along. I have my centromere following along. Here's the crossover point. I follow along and I follow along. And what happens? This is very interesting. Look, I have two reds, two oranges, a yellow, a green, and a blue. I only have one centromere, so this is going to segregate okay, but I have two duplications and a few deletions, don't I? So this piece of DNA is essentially pretty poopy. And my last piece of DNA, again, same problem here. I have a black, yellow, piece of green, and a piece of blue. So again, I have a deletion. I don't have any red and I don't have any orange. I have a centromere, so it's going to segregate okay. But again, not a piece of DNA I'd be happy to get in my cell. How about you? So this is a pericentric, this is what happens during segregation of a pericentric inversion. You end up with one cell that has the completely normal piece of DNA, one cell that has the complete DNA complement but has the inversion, and then you end up with two pieces of DNA that contain deletions and duplications. All right, the final chromosomal rearrangement that we want to talk about is translocation. And a translocation is the movement of genetic material between non-homologous chromosomes. And this can be either reciprocal or non-reciprocal. So non-reciprocal would be the movement of chromosome material from one non-homologous chromosome to another. So it could be as simple as something as simple as this. In reciprocal translocation, what would happen would be an amount of DNA would be moved from one chromosome, and then another piece of DNA from the other chromosome would go. So that in this case, this amount of DNA from this chromosome moved to this chromosome, and then this amount of DNA was transferred from this chromosome to this chromosome. So this would be considered a reciprocal translocation. A really specific type of translocation occurs often, and this is called a Robertsonian translocation. A Robertsonian translocation occurs between two acrocentric chromosomes. And when this translocation occurs, it occurs between two acrocentric chromosomes. And when this happens, it forms one very large metacentric chromosome. Now, the other fragment that forms oftentimes will contain one centromere, but sometimes this piece is so small that it actually gets lost. And what you end up with is one very big metacentric chromosome. This is actually how it is believed that humans may have evolved. And if you look at Giemsa staining of human chromosomes and compare it to the apes, you'll see that our human chromosome number 2 has staining patterns in common with two different chromosomes found in apes, and it's believed that chromosome number 2 actually may have been formed from a Robertsonian translocation.